Okay, this is what I was doing here. Stem cell differentiation is regulated by multiple signaling events. Recent technical advances have revealed that differentiated cells can be reprogrammed into stem cells. Um, Different stem cells can be reprogrammed into stem cells. The signals involved in stem cell programming are of major interest in stem cell research. The signaling mechanisms involved in regulating stem cell reprogramming and differentiation are the subject of intense study in the field of life sciences. In this review, the molecular interactions and signaling pathways related to stem cell differentiation are discussed. Okay, so this is really important. Stem cell signaling differentiation, genome reprogramming. Signals in stem cells are regulated both genetically and epigenetically by molecules. The programming of stem cell signaling is an important aspect of understanding stem cell phenotype transitions and functions. The differentiation process, as well as intra and intercellular signaling of stem cells, as described in this article. The epigenetic regulation of these cells is also discussed. Stem cells are the source of all other cells, and they have the potential to differentiate into many types of cells that contribute to the various organs, such as heart, lungs, liver, and blood. There are many kinds of stem cells. Totipotent embryonic stem cells found in embryos, mesenchymal stem cells that give rise primarily to bone marrow, stroma, adipocytes, and cord blood, epithelial stem cells that reside in the intestine, induce pluripotent stem cells that are artificially reprogrammed from differentiated cells, and cancer stem cells. In this article, signaling stem cells and the role of signaling molecules in differentiated reprogramming of stem cells are discussed. Stem cell differentiation. Stem cell differentiation is tightly regulated among the various pathways related to cellular differentiation. Epigenetic regulation is the key mechanism that controls midbrain dopaminergic neuron differentiation. Urocortin, which is expressed in the developing ventral midbrain, mediates dopaminergic neuron differentiation via the upregulation of acetylated histone H3 and the dopaminergic regulators NER1, FOXA2, and PIDX3. Dimethylcyphosphide downregulates the pluripotency gene OCT4, also known as POU5F1, and NAN-OG in human embryonic stem cells during definitive endoderm differentiation and controls hepatic differentiation. The previous report indicated that N6-methyladenosine transferase, methyltransferase like 3, regulates murine naive pluripotency. mRNA methylation is key to RNA modification and an essential factor in epigenomic regulation as cell fate determination. Pluripotent stem cells are affected by environmental factors such as cultural conditions, and such factors can affect the spatial regulations of transplanted stem cells. Several signaling pathways are involved in transformation of stem cells, such as fibroblasts, growth factor 2, FGF2, activin A signaling, and histone and lysine 4 trimethylation alterations, matrix bound heparin sulfate proteoglycans are needed for the differentiation of stem cell derived endodermal cells into airway epithelial cells. The three-dimensional extracellular matrix scaffold is essential for effective functional expression of lung epithelial cells, and HS proteoglycans also appear to play an important role in this process. Human ES cells differentiate into osteogenic cells, which can be identified by RUNX2 expression. Monitoring the osteogenic differentiation of human ES cells is useful for the therapeutic strategies used in bone regeneration. The three-dimensional polylactic acid scaffold culture of human ES cells on a nanofibrous matrix results in enhanced osteogenic differentiation. RUNX2 expression in nanofibrous matrix culture is significantly higher than in control cultures. Additionally, an efficient method for the osteogenic differentiation of human ES cells using primary bone-derived cells have been reported. Some cellular factors from bone-derived cells may be involved in human ES cell differentiation. Cell surface proteins play a role in stem cell culture differentiation. The use of functionalized hyaluronic acid surface with either